Thank you, thank you. I was going to say that I didn't recognize Carl in the suit because usually I see him in shorts running around Brookline. Good morning, everybody. Today I thought I would tell you a little bit about some of the projects in my lab that are in the process of translation, and also provide you with the paradigm that I've been trying to establish in my laboratory, which is really based on not just science for the sake of science and curiosity, but science for the sake of improving humanity and making a significant impact. And what we find in science, especially when we work on things that involve biology, is that 9 times out of 10 experiments fail. And so we experience a lot of failure in science. And I think when we start thinking about translating that science and trying to make a difference, the challenges grow exponentially. And so what I've learned is that solving medical problems is just incredibly challenging. And what I think is important to consider is that often what happens is we just run out of good ideas. We really encounter insurmountable barriers, at least they seem insurmountable, and we try to figure out new ways to approach this. And I think what ends up happening is, based on the education system that we've all been subjected to, that we've actually trained to think very narrowly about things. And when challenges arise or problems, we end up facing them, we attack them the same way every time, yet we expect different outcomes. And how many people have actually seen this TED Talk by Ken Robinson, who talks about that everybody's born creative, and the education system actually educates you out of being creative. So it's one of the most watched TED Talks, I think it's like 30 million times. And that really resonates with me, because I think that I really think that our brains go into these this super efficient mode, this repetitive mode, and it actually negatively affects our ability to solve problems. And just to kind of emphasize that point, I wanted to show you this example. So here, there's a car that's fallen into the water, and you have the problem solvers that are lined up to try to solve this problem. And they've chosen the crane here. And things seem to be going well until this happens. And so they've encountered a problem, which is a pretty big problem, bigger than their first problem. And so, because they're trained to, the education system is has made it so they think very narrowly. What happens is that they put their heads together and they come up with this as their next possible solution. But then this ends up having the same outcome. And I think that this is something that we actually face on a daily basis, is that we approach problems the same way every time. And what we've tried to do in my laboratory is try to figure out, how can we step outside the lab and think completely differently? How can we bring new ideas to to break free from that repetitive thought process? And one of the ways that we do that in my laboratory is turning to nature for inspiration. And it's this idea that every animal, every creature, every living thing out there is here because it has solved an incredible number of challenges. And those creatures that haven't have quickly become extinct. And so in essence, we're surrounded by solutions. And all we need to do is look outside, and we can bring in completely new ideas to our research to try to overcome these challenges, which are critical to solve for translation. So I'm just going to tell you a quick story about how we've tried to employ a bio-inspired approach. And this is going back to 2009, summer of 2009. Dr. Pedro Del Nido, chief of cardiac surgery at Boston Children's Hospital, sent me this email basically saying that he was treating kids who have these septal defects, which are holes in between the chambers of the heart. So nothing works. We can't suture the tissue. It just tears. There's devices that work okay in adults, but they're permanent. They're non-degradable. And the children's hearts are growing, and we can't just downsize them. This is not a good solution. So can we work together to try to solve this? And so what we did is we looked to the clinic. We said, is there anything available right now that we could tweak or use as is? And we see this product, which is Omnix, by J&J, and what it's used for is when you have a defect in a blood vessel, you clamp the vessel and then you apply this around the suture line to accelerate the time to anastomosis. So you can't close the patient up until the leak is stopped, and something like 30–40% of the patients end up having leaks. So this product accelerates that, but if you look carefully at the product insert, you'll see that it says, biological fluids and residual moisture must be removed from the entire circumference of the anastomosis prior to the application and maintain moisture-free for 2 minutes. So the tissue has to be dry when you put this on. And then it basically goes on to say that if you apply this in a wet field, it's going to attach to the fluid and not to the tissue. And so I think this material clearly has utility in the clinic for certain applications, but is not going to work inside a beating heart, which is potentially the harshest environment inside the body. And so what we like to do in most of our projects is put together a series of design criteria for the solution. And to me, this is one of the most challenging things that we do. Often it's easy to come up with a list of 20 or 30 things, but how do you pick 4 or 5 and then narrow it to 1 or 2 that's going to lead to a completely differentiated solution? So we came up with the following. We want this to be placed inside the heart and not be fouled by the presence of blood. So we want this to be able to stick to the tissue very strongly. Also, we want it to be fully degradable, and we want cells to be able to migrate into it and onto it and replace the tissue replace the material so that patient's tissue closes the hole. So we don't have to worry about that permanent implant and the growing heart problem. And we want it to resist washout because there's a lot of shear stress in the heart. Things can easily get washed away. And then a number of clinicians we spoke to said, it'd be really great if we could control the adhesiveness. So we don't want it to cure in 1 minute or 10 minutes. We want to cure it when we're ready. So we had materials that could address many of these, but not these two. We didn't really know how to address resistance to blood and resistance to washout. And so we turned to nature for inspiration. We said, what creatures exist in the sea and on the land that exist within wet, dynamic environments? And we looked at sandcastle worms, slugs, and snails. And these creatures all have these viscous secretions that stay put, like honey on a plate. And then if you look at those secretions, they also contain hydrophobic agents, which can repel water. And so we said, well, what if we just made a completely hydrophobic material that when you put it inside the heart against the tissue, it would just repel the blood away. And if it was viscous, it would stay in place long enough for the clinician to cure it. And so we developed a material that could address all the design criteria. And I'm going to show you one example here of a really challenging animal model that we demonstrated this could work in. So here we're making a 2mm defect in a rat heart, and we have this purse string suture, which is just closing the hole so the animal doesn't bleed out during the procedure. And you'll see we don't wipe away too much of the blood. And we've developed also a patch that's fully degradable, also elastic and transparent. And we apply the glue to the underside of this patch. And we're going to shine the light through the first biodegradable material, which is transparent. It's going to cure the glue. But something actually went wrong with this specific experiment. Because the heart was beating so fast, the patch we used was too small. It actually slipped away from us, so it attached. But you see this big leak here. So we had to act really fast. So we scrounged up the glue as much as we could find. We put it onto a spatula, and then we applied it directly to the leak, hoping that it would do something. And you can see it's repelling the blood. See, it's kind of clear here. So it's doing exactly what it was supposed to. It repelled the blood. It's viscous. It stays in place long enough for another pulse of the light to cure it. And then we end up with a perfect seal. And we took these animals out 6 months, and they did fine. I think we're up to over 30 animals. And so then we went to an even more challenging experiment, which is, can we attach a patch inside the heart? So this is a pig heart. We did a small incision in the myocardium, and Dr. Del Nido developed this device, a cardioport. We put our patch on the end with the glue, passed it through, pushed it against the septum, and if you can just play this video here, you'll see this is immediately after the procedure. We have 2 pigs, and the patch is attached. And then if you play this video here, after 4 hours, we add epinephrine to increase the heart rate, and the patch remains attached. And then we come back at 24 hours, the patch is still there, and we see a small capsule that's forming over top of this. And we've showed that this material can seal the carotid artery in pigs. This can attach to intestine tissue, attaches well to bone. And so to advance this, we thought, this application is probably a really high bar, but there's a lot of applications that could be accelerated. And so we formed a startup company called Gecko Biomedical, and we announced a Series A of $11 million about a year ago. And in that time, the company has fully industrialized this material. They've scaled it up, they've stabilized it, and if all keeps going as well as it has, we could be first in human sometime in September or October of this year. And I was asked to comment also on just innovation and some of the things that I've learned in the process. And I'd say one of the things that I've learned in this process is that we are just one step in the whole process of innovation, and that you need just as creative people, hardworking, intelligent people to really help with all the other steps that are involved. And so we're like one of 10 things, and each one is just as important as the next. And the other thing that I've really learned is that having innovation really requires an exceptional entrepreneur, somebody who has contacts that can help us think through all of these things immediately to really try to maximize translation, accelerate timelines. And I've had the great fortune of working with a number of great entrepreneurs. So with Gecko Biomedical, Christophe Bancel is the CEO, and Bernard Gilly is the chairman of the board. I've also had the pleasure of working with Jacques Delort, another exceptional entrepreneur, and we developed this technology in my lab in the dermatology space. We published this paper in 2011, and we've been able to launch 4 products on the market about 3 or 4 months ago in the dermatology space, and the company's called Skin2Feek. If you're interested, I'm happy to talk more about that after. And then also, we're now in the process of trying to advance another technology, which is this technology to make button batteries safer. So there's 3,000 to 4,000 accidental ingestions of coin cell batteries each year in the US. There's 33 or 34 deaths on record. And what happens is kids swallow these button batteries because they think they look like candy. They get stuck in the esophagus. And then what happens is you get a current, you get hydrolysis of the water, and you basically burn a hole through the esophagus. And so we developed a coating that doesn't conduct. If you swallow the battery, this is in a pig, we put our coating on it, so it doesn't conduct. But when you put it into a device, there's always a spring there, and that spring provides a force, and it converts our coating to a conductor. So it works well in a device, but if you swallow it, the forces aren't strong enough to convert that to a conductor, so it remains as an insulator. So thank you so much for your time, and great pleasure to be here and speak to you. Thank you.